Hi class, welcome to the Chapter 4 lecture. So the topic of Chapter 4 is on metabolism, so we'll talk about energy with that as well. Quite a few of the figures that I've included in the slide set for this chapter, they come from another textbook. So they come from Campbell Biology, and I've got the reference in here for you. Okay. And I use them from that textbook, from the Campbell Biology textbook, because I think they're a little bit simpler, but still contain the information that you need. All right, so we will cover all of the content in Chapter 4. Hopefully some of it sounds familiar once you take a look at it again. All right, so before we dive into energy transformations at the cellular level, right? That's what cell respiration is, the transformation of energy, or metabolism at the cellular level. Let's broaden our perspectives a bit. We can think of the sun as the ultimate source of energy. And it, of course, provides energy in the form of that radiant heat that plants are able to utilize. And they use this energy in the chemical reactions of photosynthesis. So that utilizes carbon dioxide and water that comes up through the root systems with that power from the sun to create a sugar. So the plant is creating glucose from carbon dioxide and water with that sun energy. So the plant's creating a biomolecule, right? We're talking about a carbohydrate, excuse me. Now, some of the energy, of course, that comes from the sun it goes into the environment. It's not by any means all utilized, but anywhere close to that. So the topic that we're going to talk about today really focuses on this side of the figure here, where we're going to review the transformations of energy that occur at the cellular level. And here, the reaction begins with glucose and oxygen. So we've got this inverse relationship between photosynthesis and cellular respiration. I'll point that out a little bit more as we go through it. So what's energy used for? Yes, it is used to perform, physical activity, to perform work, but there's other things that energy is needed for. There's also energy that's needed for chemical work, and that would be making and breaking the chemical bonds. And then we have a need for transportation related work. So the movement of different materials within a cell, or from one cell to another, or to any other part of the body. So that would include the various ions that are involved, as well as our bigger biomolecules, our proteins, our lipids, and everything that our cells and body needs. So we're going to spend quite a bit of time still continuing with our conversation on concentration gradients, since that is used right to power work. We talked about the examples of utilizing concentration gradients that are generated by one chemical reaction to help power another in our previous chapter. Okay, we've also got mechanical work, such as moving various organelles or the movement of cilia or flagella. Those are propelled by ATP. They don't just move randomly. They need energy to do that. Another example that is very relevant here in physiology is the contractions of our muscles. And we're not too far off from studying that. We'll study the nervous system first, and then we'll move into the muscles. And we're actually going to look at every single step involved in the generation of muscle movement. It's not just, it truly is movement at the cellular level, at the molecular level. So energy, of course, we have two main forms. We've got kinetic energy, and kinetic energy is always associated with motion. And then there is potential energy, which is in the form of the chemical bonds and concentration gradients. So another way to think about these two forms of energy would be, let me just grab my whiteboard over. If we're standing at a, say there's a hill, and we're say we're standing at the top of a hill holding a ball, very crude example here. And so at this point, this is, you know, the person's stationary, the ball's not moving. Let me see if I can get a better angle. There we go. So this is all about potential energy. If we're talking about the movement of the ball, it's not doing anything if the person's standing there holding it. But if the person drops the ball and it rolls down a hill, then the potential energy converts to kinetic energy, which is movement. So within our bodies, our potential energy is stored in the chemical bonds and concentration gradients that are generated through various chemical reactions and movement. Okay. So here's that example. So I was talking of, we'll focus on B, a ball at the top of a hill. There's no kinetic energy here yet. It's all potential energy. But if the ball is released, then that potential energy converts to kinetic energy, which is the energy associated with movement. This is important too, right? Going uphill is kinetic energy. And you know from doing this yourselves, walking uphill, especially if you're carrying a weight or something like that, can be, is quite a bit more challenging on our body than it is going downhill. So the difference between kinetic and potential energy. So thermodynamics is the study of energy, and you're probably familiar with the first law of thermodynamics, which is simply that that energy is conserved. So the amount, the total amount in the universe, the total amount in a system is constant, but it may change form. Our second law of thermodynamics talks about, if we want to put it into a physiological perspective, it's the amount of energy that our body uses to maintain order. So, so when we're talking about our chemical reactions, there's a lot of our chemical reactions, and we're going to look at some examples here, they need energy in order for them to even happen. Even if they may yield energy, it still takes some energy to get a lot of our reactions started. So again, in our body, we store energy in the form of our chemical bonds and then also those concentration gradients. And we're going to become quite intimately familiar with the dynamics around that cell membrane. We talked about that in chapter 5, but we're going to continue that discussion all semester long because it is the method of communication for our body. All right, so our energy can change forms, but we don't lose it. All right, so let's talk about different types of chemical reactions. We've got two main types. The first I'll introduce you to is the exergonic. So exer means to exit or to release. Exergonic reactions are chemical reactions that produce energy. So there's energy left over at the end of the chemical reaction. Oppositely, the endergonic reactions, these need energy. So photosynthesis is a great example of the endergonic reaction, and that needed energy comes from the sun. So in many times, and specifically in the chemical reactions of metabolism, we actually see a coupling between these two types of chemical reactions, where the leftover energy from the exergonic reaction is fed in to the endergonic reaction that needs energy in order to happen. And we see that in the form of the oxidation reduction chemical reactions, which are abbreviated as redox. Don't worry, we'll review them. So what we're really looking at over the distance or over the time of chemical reactions, especially big ones like with metabolism, I mean, really that's the sum of dozens of different chemical reactions that are happening. Really what we're looking at is the amount of free energy, the net energy that is that may or may not be there. And that is associated with the reversibility of a chemical reaction. All right, so let's look at that. Okay, so on the left side, we're looking at the exergonic. On the right side, in opposite, would be the endergonic. In both of these graphs, the x-axis is time, so increasing in time. And then the y-axis is the free energy, and it too is increasing along the x-axis. We'll begin with the exergonic reaction. So exergonic, let me just get a little text box here, please. Exer to exit. So in the exergonic chemical reaction, oh my goodness, sorry about that. In the exergonic chemical reaction, we begin with a level of energy that is higher than that is associated with the products. So another way of thinking about this is our reactants have a higher energy than do the products. Let me draw that in a different way on the board, please. Okay, so in the, sorry, let me turn that off. In the exergonic chemical reaction, there is exiting energy. So this means, another way of representing it, still the same axes here. Time is on the X and energy is on the Y. The reactants have a higher energy level than do the products. So that means that there is going to be the difference in energy between the reactants and the products is going to be energy left over. So in the exergonic reaction, there's leftover chemical energy that can be utilized for another reaction. So in contrast, the endergonic, I remember that for enter. It's the opposite situation. Here, the reactants have a lower energy than do the products. In other words, there is no way to make the products of an endergonic reaction without an input of energy, and it would be equal to the difference between them. So this is the coupling. The leftover energy of an exergonic reaction is used for an endergonic reaction, since the endergonic reaction needs it. So the way I like to also think about this is, I mean, similar to like a bank account, you have to have money in it in order to spend it, but we don't want to get stuck up on the specific chemical reactions. It's not like we can say that chemical reaction exergonic A always is associated with endergonic reaction B. It's more the energy totals that are in a cell. So the cell is going to build up a bank of energy from its exergonic reactions that then can be utilized to power endergonic reactions. That's a common misunderstanding I've seen, is that it is not realizing that it's really banked. The energy is banked for a period of time. That's a helpful way to think of it. Okay, let's get back those slides. All right, so moving forward. Okay, so that's kind of what I was just talking about. We can visualize here. So we have our exergonic reaction. So the products of A and B yield C and D, and there's energy that is released. So some of it is lost in the form of heat. So it's lost from this system in the form of heat, but it's still within the overall environment or system of the world. So it still applies to the first law of thermodynamics. It still applies. But some of it is passed through in the form, as we're going to see, of energy, high energy electrons, and that brings in the role of some of our electron carriers. That's what I like to call these molecules: NADP, NAD, FAD. They've all got the H on there right now, so they're carrying their electrons. But these electrons can be shuttled then to produce ATP, which then can go in as the source of energy of the endergonic reaction. So see how there's the banking of the ATP in the middle here. So it's not that you need A plus B to give you G and H. It doesn't work that way. We're just making energy, and then the cell can use it for whatever chemical reaction it needs. Okay, I hope that helps to clarify that. All right, so let's talk about enzymes. So having an enzyme, it does something. It often, the way to help think about it in our body is that it makes the chemical reactions possible. So strictly speaking, an enzyme is a biological catalyst. That just means it's a catalyst that our body makes. It's an organic molecule. 99% of the time they are protein, but there are some examples made out of RNA. So what they do is they they decrease the barrier that's needed to make the products. So we don't call our starting material a reactant when there's an enzyme involved. It has a different term. They're at that point called substrates. So if you see the term substrate, automatically you should be thinking, oh, that means there's an enzyme that's required for that chemical reaction to occur. And in fact, the chemical reaction won't occur properly without it. Okay. So there are some examples in our body of what are called isozymes. Iso means the same, right? We've seen that prefix before. We saw it in the example of an isomer. But an isozyme, these are enzymes that under different conditions, different enzymes that under different conditions can work in the same chemical reaction. So what that effectively translates to in our body is our body can make one protein, one enzyme, and under different conditions, it can be used in more than one reaction. So that means that's less material for the body to have to make. It only needs to make the one enzyme instead of two. Okay, so now let's start talking about some of the variables that do affect the rate of reaction. And temperature certainly is one of them. The way that temperature works though, I mean, it's kind of a double-edged sword with enzymes. What temperature does is it increases molecular motion. Things move faster. And that does mean that if two substrates need to come together to make the product, that there's a higher chance that they'll bump into each other if the temperature's warm, but not too hot. The other side of it is that heat can actually destroy an enzyme. I mean, heat can make anything that has hydrogen bonds can fall apart. So, so we have to think about temperature within a range. I mean, that's really true of any variable. It's always within a range. There's extremes. But varying the amount of substrate or even the amount of enzyme can also affect the rate of reaction. Now, many of the reactions of our body will hit equilibrium. And that tells us they're reversible. So our reversible reactions are going to hit that state of equilibrium where they're balanced, not equal, but balanced. And then that means they can go the other way. I'll show you a clear example of this in just a moment. I've got a nice, pretty chemical reaction type for you instead of having to write it for you. Okay, so let's also talk about some of the other dynamics or kind of factors that affect enzymes. So enzymes are proteins, and proteins take quite a bit of time to make. They have to go through their 4 layers of folding: primary, secondary, tertiary, and quaternary folding before they've got their right structure, which then gives them their right function. But what if we need the enzyme quick? Our body has a way of dealing with that. It makes them in what is called a pro-enzyme. So when you see this prefix pro in front of any protein, it means that the enzyme is produced, but it needs to be cut. It needs to have one quick cleavage done to it, and then it's active. I'll show you examples. Coenzymes, just so that we can run through the vocabulary, and then we'll look at examples. Coenzymes are, these help an enzyme, right? We've seen this with other examples. But where do they come from in our body? So many of them come from the vitamin Bs. So that is the thought and perhaps the main science, the main chemistry that drives all of those, I don't know what the proper term is, but you can go and get an IV that has vitamin Bs in it to get energy. There's a bunch of places like hydration bars or medical spas offer these. But what that is it's an infusion of the precursors that are needed to make the enzymes that are needed for respiration. I'm going to point this out on the board. I'm going to draw some of this out for you. So we talked about how temperature affects enzyme rate. pH has the potential to destroy an enzyme, as do other chemical factors. Basically, an enzyme needs to work in the it needs the physiological conditions. So most of our enzymes are going to work at our physiology. So they work by lowering what's called the activation energy. So they're not affecting this. Endergonic and exergonic reactions they still are associated with the net ebbing change of free energy. They're not associated with the activation energy. Let me please show you what that is. Okay. So in this case, we are looking at what would be the exergonic reaction, just so that we can apply what we've already learned. But the thing about energy is this barrier here. So look at this bigger red line versus the shorter red line. Kind of wish your book used different colors. I could call it out easier. But anyway, the way that we read this is without an enzyme, substrates A and B need to get up to this high of a level to form the products C and D. And oftentimes for physiology, what this means is the reaction's just not going to happen quick enough. Usually the reactions will favor the formation of products if we've got the right quantities of everything, even without the enzyme, but it's going to be too late. It's kind of like how, let me think of another good example. Oh, it's kind of like talking about glucose being a non-penetrating solute. Technically, it's penetrating. It just takes so long that we can't consider its effects when we're looking at other solutes. It's kind of like this. The chemical reactions it will occur, but everything else is going to be over and done with, and it's just irrelevant. But having the enzyme present, what it does is it reduces this enzyme barrier called the energy of activation so that it is easier to form the products C and D. Now, one thing I haven't mentioned yet that is a very important part of understanding enzymes is they're not used up or consumed in the chemical reaction. They're free to go again and again and again. The enzyme is not part of the reactants, all right? It's something that simply helps to decrease the activation energy. And I'll show you a good example of how. Let me just erase my board real quick and we'll go over that. Okay, now you can see better. Oh. Sorry, I'm getting tangled up in books and chairs. Okay, so many times what happens with our enzymes, this is called a type of enzyme called a substrate level enzyme. So here it is. This is our enzyme. Okay, I'm a terrible drawer. Again, kind of looks like some weird animal, almost like a dinosaur to me. Okay, so what it is we have A, and then we have B. Okay, let's get it all drawn out. So this would be the substrate example of an enzyme. We have our two substrates, A and B. This is the enzyme. What a substrate enzyme does is it simply holds the reactants in place. So A is going to come in here into its pocket, and notice B has a different shape. So it goes into its pocket, and then with the right chemistry, meaning all the other chemicals are correct, we generate the product, which now has a chemical bond that links A and B together. Okay, so that's an example of the substrate level enzyme. Substrate level phosphorylation is often how you'll see those terms. The addition of a phosphate group is another great example of this kind of reaction. And so what other reaction am I referring to right now? Our energy cycle. Adenine diphosphate plus phosphate makes adenine triphosphate, which breaks down to adenine diphosphate. It's actually a cycle, and there's an enzyme that does this. So just to give you two examples now of substrate enzymes. This is important in our understanding cellular respiration because we do utilize substrate level phosphorylation in part of the chemical reactions there. Okay, let me get your screen back. So if we were in the classroom, we would turn to our nearest neighbors, and amongst yourselves, you would discuss these points. So I will leave them to you to work through on your own time. So about enzymes, you would discuss the active site, which the binding site of A is the active site for A. The binding site of B is the active site of B. The binding active site is the site on the enzyme that binds its substrate. So it's determined by what structural level. What level of folding has to happen to that enzyme, which is a protein, so that it has the correct structure so that it functions as an enzyme. I'm not going to tell you the answers. I'm just wording it for you a little bit differently. So how does an enzyme behave as a catalyst? Basically, what I'm asking you to do is prove how an enzyme is a biological catalyst. Could you draw a chemical reaction, either graphing it or like this, that would show how an enzyme is a catalyst? Can an enzyme be used multiple times? Or in other words, is the enzyme consumed in the reaction? Does it become part of the product? If it did, then we would run out of it as more product is made. That would severely limit the ability to make the product. If the enzyme is limiting, then we don't have enough for it. What is the name of a molecule that helps an enzyme? We just talked about this. And what are the different factors that affect the rate of activity of an enzyme? All right, so let's talk about one more example before we move forward. Let's talk about the example of heat versus enzyme. A lot of people will describe heat as an enzyme, and let me give you a very clear example why it's not. Say you have a cup of water or pot of water, it's room temperature, right? It's just, you know, it's not bubbling, it's not boiling, it's just chilling in my cup. But if I put it on the stovetop, if I put my pot on the stovetop and apply heat, what happens over time? Well, there's a transfer of energy in the form of heat. That's what heat is. It's a transfer of energy from a hotter object to a cooler object. So the pot of water starts off cool, the burner gets hot, it gets hot, 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 and eventually the water is going to start to move around. And that is going to mean that the water molecules are hitting each other more quickly. The hydrogen bonds are breaking. So as water heats up, the hydrogen bonds break, and the water molecules move quicker, which means they could hit other things and bounce into a reactant, or I should say a substrate quicker. But what happens when you take that pot of water off the stove and you set it down at room temperature and you let it cool down? It doesn't stay boiling. So therefore, heat does not fit the definition of an enzyme because it is consumed, right? It doesn't, you have to keep putting it in and keep putting it in to keep the water boiling. So that's one example of why it doesn't fit it. But often, yes, it is true, heat does increase the rate of a chemical reaction. In fact, let's apply that to a physiological example now. What happens when we get a fever, and why do we get a fever? Well, one of the big triggers for our body, for our immune system to produce a fever is when it sees, through its immune system eyes, we'll learn about that, is when it sees that there's a bacteria infecting us. Bacteria live just in our body. They don't live inside cells really, but they live like on surfaces and inside our body. So when our body senses, when the immune system senses that there's a bacteria in there, it's going to elevate our core body temperature. What's that called? Well, that's a fever. Why is it doing it? Well, many of our immune system cells and the molecules that our immune system produces so that it can effectively deal with this invading bacteria, they are actually, they work better with a little bit of heat. So there is a reason why our body is producing that temperature, and it actually helps to activate our immune system, and certain parts of our immune system are more effectively activated. It also helps our immune system cells get to the site of infection quicker. Say the infection is in the skin. Well, what good is it going to do if all my bacteria killing immune system cells are in my toe? It's not going to be very effective in my skin on my arm if they're all somewhere else. So that mild increase in temperature helps the immune system cells get to where they need to go. Now, a high fever, it can be dangerous. And when I say high for an adult, you know, something a sustained 101.5 or higher, or if a person has taken a fever reducer and they're still high, that's a concern. That's when that person really should be, you know, trying to cool their body temperature down, taking a fever reducer, taking an ice bath, and if it doesn't work, then they need to go get emergency medical help. Because why? Well, a high body temperature is going to denature our enzymes. What's it doing to the bacteria? Well, exactly that. It's trying to denature the enzymes of the bacteria, which is good for the bacteria to get killed. But it can also do the same thing to us, because the bacteria that infect us, they've evolved to live at us conditions. So they too live at our good body temperature. You know, they've adapted to live at 37 degrees Celsius on average. So that's another example of understanding the role of enzymes. So many of the chemical reactions of our immune system are quicker and because of heat, but that is not an enzyme. It just makes things move faster. Okay. Yeah, so try to draw that example out yourself. Draw a pot of water at room temperature and what it looks like boiling, and then what it looks like back at room temperature, and even what it looks like when it's frozen as ice. And when I say draw it, please do it at the molecular level. You know, draw your water molecule and show how it hydrogen bonds to the other things that are present in your solution at each of those phase changes. All right, back to our lecture slides. So enzymes can be categorized by the type of chemical reaction in which they're doing. So phosphorylation is a big one, and we're going to see it over and over, because ADP is the addition of phosphate to adenine diphosphate, that is phosphorylation, the addition of a phosphate group. That's all it means, is phosphorylation. You are phosphorylated if you add a phosphate group on. So we have a bunch of enzymes that function this way, but we also have these redox reactions, and we need to understand the role of enzymes here, and specifically the role of the coenzymes as well. Oh, I'm jumping ahead. So when we say something is reduced, oxidation is loss. Let me get my text box. Excuse me. There's an acronym we can use called oil rig. Oxidation is electron loss. Reduction is electron gain. Oil rig. Oxidation is loss. Reduction is gain of electrons. Okay. So we're going to review those because that's metabolism for you. And we've already talked about the hydrolysis reaction and dehydration synthesis reactions that are used to either make a polymer or break a polymer. So in our dehydration synthesis reactions, we're making a polymer. So we're starting with the beads and then we're making the necklace. Water is always a product. Dehydration, it means to take water out. And then the reverse reaction is hydrolysis, which is the addition of water. And that is also called digestion. So when we eat food, part of the digestion process is actually hydrolysis. All right, now let's get to metabolism. So please be able to define what metabolism means. And if I ask for a chemical reaction, I hope you know that. I hope you know how to write it out correctly. We'll look at it. So we've got our catabolic and our anabolic metabolism. And so metabolism is the sum of all the catabolic and all the anabolic. So catabolic is derived from catalysis, which means to break. So digestion versus anabolic means to build. Think of anabolic steroids as an example, to build muscle mass. So you're building something. So our metabolism is the sum of all of our food that we digest down in our catabolism, and then all the molecules that we have to make. So it's the sum of all those chemical reactions. All right, and we will look at the importance of redox reactions in our metabolism. We could not have metabolism without these redox reactions. Also, we need to appreciate the role of the coenzymes. These are derived from vitamin B. They are the enzyme shuttles. So let me just take a moment and draw a picture of that at this point for you. I lost my pen. Oh, here it is. Okay, so we're looking at an example of a coenzyme. And again, these are made from vitamin B. So we're going to look at the example of NAD. So we have different forms of NAD. So this is the oxidized form of NAD because it's got no electrons. Oxidation is loss. But if we take NAD and add 2 electrons to it, what does that produce? It is going to reduce it. But let me show you a useful picture way of looking at it. So NAD, I like to draw like this. So this is NAD plus, meaning it's oxidized. It has no electrons in its electron pockets. So I draw NAD like this because it's easy for me to see. So NAD plus 2 electrons becomes just bear with me, please. Sorry, I should have done these in a different color. Okay, electron 1, electron 2. And now we are reduced. NADH is reduced form. Okay, so this is an example of actually a couple good points we need to learn. It's an example, this is an example of a redox reaction, which is abbreviated REDOX. So it is a good example. So this is the reduction. Reduction is gain of electrons. But if we take the electrons out, if we minus those 2 electrons out, if we shuttle them into the electron transport chain, then our NAD is oxidized and is properly called NAD+. That means it's oxidized. So NAD+, 2 electrons creates NADH. A lot of times and even I did this when I was a student and I didn't understand it. I wanted to call this NADH2 because it's like, well, we got 2 electrons, so why on earth aren't we writing a 2 there? Well, because of this plus. So the plus sign takes the first H and then the second H shows here is a way to think of it. All right, so this is a coenzyme. This NAD thing that can exist either oxidized or reduced is the coenzyme, and it's showing the redox reaction, the transfer of the electron. There's also other molecules. We've got NAD, but we also have another one called FAD plus, which is oxidized. And then there's another one called NADP plus. The way I think of this one, it's not always true, but many organisms that use NADP as their electron carrier, it's going in the chemical reaction of photosynthesis. It's not always true, but a lot of times it is. So we're going to talk about NAD and FAD primarily. Now, I did load a bunch of YouTube videos, 5 different YouTube videos in your lecture folder page for this chapter. Please watch them. I previewed so many videos trying to find good ones. That you'll see that many of them are made by a gentleman named Paul Anderson from Bozeman Science. He's fantastic. The reason I use him is because his videos are at the right level without having too much stuff. And I have not, actually I did find one mistake one time. I emailed him and he fixed that thing right away. So I've got to respect that. So if you need a topic and you don't know who to look up, or you're looking for a good source, I do encourage you to use him, Mr. Paul Anderson. Just search for him in the topic and you'll find it. He teaches a lot of AP biology. All right, so now we're gonna review the steps of metabolism. And we're not gonna look at every single chemical reaction of metabolism. We're gonna look at them on a stage level. And I'm gonna be very clear with the things I want you to learn, all right? Because this can get very deep and intricate, but it does not need to for your introductory physiology class. It needs to get deeper as you move into as you develop more of your knowledge. But for now, the way I'll teach it to you, I hope is an effective way for you to learn it. So stage 1 is glycolysis. There is a transition step that must happen. Next, stage 2 is the citric acid cycle. This is also called the Krebs cycle. Oh, and Charlie Krebs, the dude who invented or discovered the Krebs cycle, he was a physician at Scripps, so a local San Diego connection. His son is currently a physician in the Scripps hospital system. I'm a total nerd. I unfortunately was admitted to the hospital there. I have bad asthma, and Dr. Krebs came. He was part of my treating team, and he thought it was kind of hysterical that I knew who he was, because I was actually prepping for this lecture at the time. And then stage 3, I call the electron transport system. This is different from how a lot of textbooks refer to it. Your textbook calls it oxidative phosphorylation, which it is, totally accurate. A lot of textbooks call it the electron transport chain. I'm going to challenge you to see why it's not accurate to call it the electron transport chain. That only takes into account what happens with the electrons, which electrons are important, but when we're talking about making ATP, there's a huge role of the proton as well, and that does not get taken into account when we just limit our discussion to the electron transport chain. So within the electron transport system that uses oxidative phosphorylation, we do have the electron transport chain as well as the proton motive force, which is called chemiosmosis. That's the same thing. In fact, the person who discovered chemiosmosis won the Nobel Prize because of the recognition of how important it was. This is where we make most of the ATP, is from those protons. So this is what I want you to know for each of the 3 main stages. So I don't expect you to know all of the chemical reactions of the citric acid cycle. I mean, there's tons of them. What I want you to know are the starting materials, and we'll review this. What that stage ends with, the net amount of ATP produced for that stage, the location, where does it occur? Is it in the mitochondria? Is it in the cytosol? Is it in the outer membrane of the mitochondria, the inner membrane of the mitochondria? Which coenzymes are primarily used, and whether or not it is aerobic or anaerobic. And you'll see in the lecture objectives for this chapter, I gave you a nice table so that you can fill this information out. We've got a table in the slides as well that we'll look at. So glycolysis, citric acid, and then the electron transport system. So a cell or organism that gets through all 3 stages will make more ATP than a cell that just does glycolysis or the citric acid cycle. And even just looking within the example of our muscle, different muscles have different metabolism requirements. So we have some muscle that primarily just uses glycolysis, but then we have other muscle that uses all 3 stages. And the amount of ATP produced is very, very different. So there's got to be some benefit to producing less ATP, or else it wouldn't happen. We would have, things would have gone differently over the thousands of years of evolution, millions of years of evolution. All right, so this is the balanced chemical equation for cellular respiration. This is something that you'll need to know, and you'll want to be able to retrieve it quickly, or make yourself have it out in a nice place that you can quickly get to. So let's review it. So we begin with 1 molecule of glucose, which has 6 carbons, 12 hydrogens, and 6 oxygens. It can exist as a ring, like we're showing here. Glucose can also exist as a straight chain, but we'll leave it as a ring. It reacts with 6 molecules of molecular oxygen, O2. Oxygen bound to oxygen. That's molecular oxygen. And that's what exists in nature. It's not correct to say O, because, well, that's the oxygen free radical. It doesn't just exist that way in nature. It would bind to something positive. So we do need to keep track of our correct materials here. 6 molecules of molecular oxygen goes through glycolysis, citric acid, electron transport system. That's why there are 3 arrows to represent the 3 stages of cellular respiration. So at the very beginning of cellular respiration, we have glucose and oxygen. After all 3 stages, we have 6 molecules of carbon dioxide, 6 molecules of water, and about 30 to 32 molecules of ATP. And some energy is lost in the form of heat. So that is the balanced chemical equation of cellular respiration. Let's address a few things. Heat. Yes, so there is some energy that is released. That means it is not associated with the chemical reactants or products. It's lost to the environment of heat. But we're talking about cellular respiration. So the environment is the rest of our body. Our body is smart. It utilizes this leftover energy in the form of heat. It is part of our internal body temperature. So when we're cold, you know what happens, right? When we're real cold, you shiver. What's happening? Well, your muscles are contracting. They are undergoing cellular respiration through contraction to generate heat to increase the body temperature and get it back to its normal set point. That is what shivering is, and it is triggered by cold. So we utilize the heat. Okay, so let's pause a moment and look at the chemical reaction, the redox reaction. My advice is always to follow the hydrogen and the carbons of the chemical reaction. Let me show you why. Oops, I just scared the kitty here. Sorry, Meatball. So we follow the carbons because, well, the carbon is the backbone of all of our biomolecules. So that might be a little bit easier to understand why we follow the carbon. So we have C6H12O6 plus, oops, 6 molecular oxygens produces 6 water, 6 CO2. So we're going to lose our, let me just draw this out. Oh, sorry. Let me turn off the screen share. You can see better. I apologize for that. Okay, so we've got our balanced chemical reaction of cellular respiration. 1 glucose molecule, 6 O2s, 6 waters, 6 CO2s. So just to review the balanced equation, on our reactant site, we have only carbon, hydrogen, and oxygen. Over here, we have carbon, hydrogen, and oxygen. So we're essentially demonstrating the first law of thermodynamics, that matter is not created or destroyed, but we're also checking the balanced equation. So we have 6 carbons. We have, this is 12, 6 times 2 is 12 oxygens, plus we have 6 here. So there are 18 oxygens, and then for the hydrogens, there are 12. So for it to be balanced, we have to have the same number on the product side. We have 6 carbon. We have 6 times 2 is 12 oxygens, plus 6 is 18. And then we have 12 hydrogens, and it's balanced. Okay, but now let's get back to that redox thing. So I follow the carbon and the hydrogen, and I always recommend to my students to do the same thing. Hopefully by just going through that example of balancing the chemical equation, you can appreciate why we follow carbon or any of the elements, but we're following carbon because it is that backbone of the molecule, right? When we looked at our glucose ring, every point is a carbon, the backbone of the molecule. But why am I telling you to follow hydrogens? Well, hydrogens with their atomic number of 1, that means there is 1 proton, which we're going to abbreviate by H+, and that means there is 1 electron because it has an atomic number 1. And we're talking about covalent bonds if we're talking about building the biomolecule or breaking it. So we follow the hydrogens because this is the source of material that goes into the electron transport system as well as the citric acid cycle. So going back to our overall equation, if we're trying to figure out, well, which molecule, if I asked you, which molecule is oxidized or which molecule is reduced, please don't tell me water or carbon dioxide. Those aren't even options. Because if I'm asking you which molecule is reduced or oxidized, it has to be a reactant. So if I gave you, a choice for 4 choices here, 2 of them are already wrong if they're in the reactant side. But how do you determine which is oxidized and which is reduced? Well, we've got our acronym. Oxidation is loss, reduction is gain. The molecule so oxidation is loss. So the only molecule that even has hydrogens, so we're thinking of electrons for oxidation or reduction, is glucose. So glucose can give away some hydrogens, which would allow something else to be reduced. But what we're seeing, what we see here between going from glucose to water actually, I drew that wrong. That's why I'm tripping over it. I'm so sorry. Let me correct my connections here. I wrote the reactants up differently in order from how I had them listed. So the relationship here is between, one of the relationships is between oxygen and the water. So oxygen can accept electrons. It would gain electrons. So it is reduced into water. Oxygen picks up hydrogens. It's gaining. Oxidation is loss, reduction is gain. It is reduced to becoming water. Whereas glucose gives away, it's got the opposite connection with carbon dioxide. It becomes oxidized. So the molecular oxygen actually gets the electrons from glucose and is reduced into water. But when that happens, when glucose gives away its hydrogens, it becomes carbon dioxide, right? If we take away the hydrogens out of glucose, we're left with carbons and oxygens. So that is the relationship between the transfer of electrons, but it is tied to why I advise you to follow the carbons, so you know the size of your different biomolecules, and you can balance it if you need to, because if it's not balanced right, your answers would be wrong. But then we follow the hydrogens so that we can figure out which molecule is oxidized and which molecule is reduced. So oxygen, molecular oxygen, is reduced into water, and glucose is oxidized into carbon dioxide. All right, so let's move forward So here's the sum of all three stages. And this would be aerobic cellular respiration because we're going through all three stages. So it begins with glycolysis, which is stage 1. We have our transition step. Then comes stage 2. Electrons and protons are shuttled away into the electron transport system where a lot of ATP is produced. So the bigger the star around the ATP, the more ATP that's there. This figure is also showing you the location of each stage. So by the time the reactions progress from the transition step through the citric acid cycle, through the electron transport system, all of that is happening in the inner mitochondrial membrane, right? That's the one that has all the wiggles. More wiggles means more cellular machinery for cellular respiration. So that's why that membrane has all the wiggles, so that there's more space and more equipment to carry out the reactions of cellular respiration. So let's look at a couple other things. So the oxygen isn't utilized until stage 3, which is why it's called oxidative phosphorylation. The carbon dioxide is released after the oxidation of pyruvate in the transition step. Water is produced in stage 3. So the different materials, our reactants, are utilized at different stages. We're looking at the overall sum reaction of cellular respiration, as well as at each step, each stage. So glycolysis happens in the cytosol, but everything else happens in that inner mitochondrial membrane. So here's the source for this figure. They come out of that Campbell biology, which is very commonly used for the intro bio classes. So let's look at the overall chemical reaction of glycolysis. So this top figure is just showing us really a map of the whole process of cellular respiration, and it's showing us what part we're talking about. So glycolysis begins with 6 carbons in its glucose. So we've got 1, 2, 3, 4, 5, 6. It is broken down, our 6 carbon glucose is broken down into 2 molecules called pyruvate, and each of them contains 3 carbons, right? So we've got 3 plus 3 gives us our 6 carbons that we need to track. It is utilizing 2 molecules of NAD plus to shuttle its electrons in the chemical reactions. So we produce 2 molecules of NADH. We utilize 2 molecules of NAD plus, and we make some ATP. Only a net production of 2 molecules of ATP is produced. We actually make a total of 4 ATP in this stage, but 2 are utilized. So that means there's a net production of 2. So the information I'm asking you to learn and be able to work with is really represented here. Our starting material is glucose. Our ending products are 2 pyruvate, and it's very important that you say 2, or else the law of thermodynamics has been broken. A net production of 2 ATP are produced. 2 NAD pluses are reduced into 2 NADH, and it occurs in the cytosol. Now, there's a transition step that has to happen in between stages 1 and 2. And what this is it's oxidation of pyruvate. Now, keep in mind, you have 2 pyruvates. We're only looking at one of them here to keep it simple. But what happens is actually one of the carbons is lost as carbon dioxide. Our molecule here, we reduce NAD plus into NADH, And then in step 3 of the transition step, we have this enzyme called coenzyme A that comes in and joins on to the 2 carbons. Now, CoA has a carbon in there, so we technically have 3 carbons. So this is oxidation of pyruvate. So the starting material for the transition step is pyruvate, and the ending material is acetyl coenzyme A, or acetyl-CoA. There's no energy transformation that's happening here with ATP. Now we've gotten to stage 2, which is the citric acid cycle. The starting material is acetyl-CoA. All right, it's not pyruvate. It has to go through that transition step. And it is a cycle, so it goes around and around. The starting material is acetyl-CoA. We have 3 molecules of NAD+, which are reduced into 3 NADHs. We make a total of 1 ATP, and then there is also a reduction of 1 FAD into FADH. And this happens in that inner mitochondrial membrane. Now, what we need to take account of, though, is we have 2 molecules of acetyl-CoA. If we're working from 1 glucose, it makes 2 pyruvates, which means 2 acetyl-CoA. So you have to double your yields. If I'm asking you the total ATP produced in the citric acid cycle, it's actually 2, and 6 NADHs are produced, because you've got to process both molecules of acetyl-CoA. So it turns twice for every 1 molecule of glucose. So the ending product, if you're looking at a more complicated figure, gives you another chemical name here that goes right back into the cycle. The ending product for our purposes is, it's the electrons and protons that are loaded on your carriers. Because what I'm asking you when I'm talking about the starting and ending products, the ending products are what go into the next stage. So what goes to the next stage after here are the electrons that are shuttled in our carriers. Now, I want to go over one other point about our carriers before we go any further. All right, so 1 molecule of NAD+ can hold 2 electrons. So let's put our electrons in the pockets as we were doing. But hydrogen has, yes, it does have the electron, but it also has an H+. So in reality, the H+ is coming along with it, because it's not, we haven't taken the electron off. Well, we have, but it's coupled. So a way to think about it is to also represent that the H+ is coming along with it, because it is. So I like to think of it as the electron is seated like in the roller coaster, and then the proton's holding hands. So the proton can get released and go to somewhere else really easily. So we don't want to forget about our protons. So the ending materials are the electrons and protons that were loaded onto our 2 types of electron carriers, NAD+ and FAD+. And then stage 3, so this is the electron transport system. It utilizes the electron transport chain. So this is a complex, a series of different proteins. So we take out our materials out of our citric acid cycle. The electrons go into these pumps. So notice how there's an H+ being pumped through. So these are actually electron pumps. So the electron goes through the chain, but the protons get pumped and they build up in this intermembrane space. So we don't want to forget about the role of the protons. There's so much focus on the electrons, but those protons are so important. And we have 3 pumps in the series of the electron transport machinery that's in that inner membrane. 3 proton pumps. So our electrons, again, go through the electron transport chain, and now they are free to react with molecular oxygen to create water. So going back to our products, molecular oxygen picks up these shuttled electrons to become water, which is what we're seeing here. Really, this should be a 1/2 O2 to make 1 water, right? Because we have 2 hydrogens and 1 oxygen. Well, how do we start? It's not balanced. So this should be balanced. So, so we can look at, so right, it would be 1/2 O2 plus 2 H's makes H2O. One-half O2 plus 2 hydrogens makes 1 molecule of H2O. So it's got to be half of molecular oxygen in order to balance it correctly. And that sounds like it might be silly, but it isn't, because O doesn't just float around in our body or in the air without causing big problems. It's going to react because it's very chemically unstable, very high energy. So it's not going to exist that way to hang out for this chemical reaction. So we need to appreciate that this molecule right here, 1/2 O2, this is the final electron acceptor or terminal electron acceptor. It is what picks up the lost electrons that come off of glucose. They react to form water at this correct ratio. Oh goodness, I could just smell smoke again. I don't like that. All right, so that's the fate of the electrons. The protons go through this interesting molecule, ATP synthase. Well, that name's telling you a lot. The ASE tells you it's an enzyme that synthesizes ATP, and it does. Look, adenine diphosphate plus inorganic phosphate makes ATP. It varies a little bit on what type of cell we're talking about and even what organism we're talking about and the specific chemical conditions of the time. But essentially, for every proton that goes in, we're making a molecule of ATP on average. So we make a lot of energy this way. So here's the molecule, ATP synthase. So it's a rotor pump that sits within that inner mitochondrial membrane. So it has a place where it can load lots of H pluses, and then it turns so that it can, and keep in mind the H plus is hydrogen. So as it pumps the hydrogen through to the inner membrane space, it is phosphorylating adenine diphosphate to make ADP. So that's ATP synthase. So here's a table that summarizes each of the 3 steps. I did not include the transition step in here, but hopefully you would be able to insert that data in if I asked for it. The table I gave you in your lecture objectives does. But look, so these are the materials that I'm expecting you to put back on an exam or quiz. So keep in mind the ending material, what I'm looking for is the material that shuttles into the next stage. So for the end of the citric acid cycle, it's the electrons and protons that are loaded on the carriers that get put into the electron transport system. However, the products of the electron transport system, this is actually when the carbon dioxide and the water are physically made. So they are the products there. So if we add together the net production of ATP from one glucose under aerobic conditions, it varies between 30 to 32 molecules of ATP, whereas the other stages only produce a net production of 2. Now, let me back up a little bit. There are certain conditions where the citric acid cycle is going to pump the electrons in later into the system. And in fact, if we're using FAD as the electron carrier, it doesn't even work until we're already moving through the electron transport chain. So these are some of the reasons why the amount of ATP can be less and why the number of carriers can be less or variable, because it depends if we're talking about starting at the very beginning of the chain or dumping in somewhere else. And there are conditions that will make it so that we omit part of the chain sometimes. So that's why you see it varying with the electron transport chain or system. Another reason why we sometimes see some variance in the molecules produced is the cell can be utilizing them right at the same time it's producing them. All right, so this is the figure from your book. I inserted these extra figures because I think we do need to break them down a little bit. But this is important to see because it breaks out the primary active transport versus secondary active transport that's occurring through the some reactions of metabolism, cellular metabolism. So we talked about primary active transport is exemplified by the role of the sodium-potassium pump that pumps out 3 sodiums for 2 potassiums in. It uses energy because it's going against the gradient. But when it does that, it builds up potassium and sodium in their respective environments, which then allows this channel to be used. So we've got a buildup of sodium now, and that sodium gradient is going to help to drive the secondary transport of potassium and chloride following their gradients. So secondary active transport is the utilization of a gradient that has been generated by the use of ATP and a previous step in the same reaction. This is the figure from your textbook, so I included it. Just because it's what you have in your book, but this material is summarized in the table that I have a few slides back. But you can see that by far the majority of our energy comes from the electron transport system. All right, so we've talked about NAD, and it comes from niacin, which is a vitamin B3. We also have FAD, which comes from riboflavin, and then coenzyme A, which is used to make acetyl-CoA. This comes from pantanoic acid, which is one of the B5s. So we talked about the industries that are utilizing infusions of these various vitamin B molecules. The idea is to provide more of the precursors of the electron carriers so that there's no limit of the carriers in doing their job in that oxidative phosphorylation process. So the latter stages, that last stage, stage 3, is oxidative phosphorylation because it's occurring in the presence of oxygen. It's no longer substrate level, it's oxidative. All right, so what's the difference between aerobic and anaerobic? Well, if you have a cell that's capable of doing both, most of the time it's going to choose aerobic because it is so much more efficient. What happens with anaerobic metabolism is it's basically just glycolysis. But if we have a situation where pyruvate's not able to oxidize and move on to the next stage, right? To oxidize pyruvate, we need oxygen. But if we don't have oxygen, we can't do that. So instead, we go into anaerobic metabolism, and we'll look at lactate acid production as an example. But we have this problem of what happens when we load our carriers. Let's look at the figure. So remember, glycolysis begins with 6 carbons that are broken down into 2 pyruvate, each having 3 carbons. But we do reduce 2 molecules of NAD plus into NADH. Well, there's the end. There's the end of anaerobic respiration if it were not for lactic acid fermentation. So anaerobic respiration really is 2 steps. It is glycolysis plus the regeneration of the carrier. And so now let's look at this second part where we need to oxidize the carrier. So we begin with our 2 pyruvate and our 2 loaded NADH molecules. So all that happens here is the NADH molecule is oxidized back to NAD plus, and now it is free to be used again and again and again, as long as we go through this step of converting the 2 pyruvate into lactic acid. So you do generate energy in anaerobic metabolism, just 2 ATP for every 1 glucose, but also we oxidize the NAD so that it can go back and be used again and again and again. So we really can't, I can't underscore how important this second stage is. One of the YouTube videos that I loaded onto your page for chapter 4 talks about anaerobic respiration or fermentation. If you're stretched on time and that's the only one you look at it, please look at it. It's really helpful to understand what's going on here. So let's think about our body. So most of our cells do utilize aerobic respiration, but we do have some, including the red blood cell, that doesn't. So our mature red blood cells, not the immature ones, only the mature ones, they only do glycolysis. So I'm going to incorporate this into our lecture, into the discussion topic for week 5, as to why you think that there's a benefit for using glycolysis for our red blood cells, and also relating the structure of the red blood cell to this purpose. So if you want to jump ahead and read about it, you would go to chapter 16. But I think that you can probably just think about structure and function and perhaps come up with your own hypothesis on this. This is the last slide. This is a figure that comes out of the end of the chapter review from the introductory biology textbook that I use. But I wanted to put it in here because I think that they're helpful. So I suggest that you utilize this as a way to understand your ability to conceptualize what's going on here. Maybe even try it at the very beginning of studying this lesson, and then again at the end. These tend to be tricky. They take some thought. All right, I'm gonna sign off now and hopefully get this video back quick so I can load it up for you. Take care, everybody. Stay safe. Bye-bye.